Department of Biotechnology of Atmia University welcome you all in the virtual lab session on preparation of Murashige and Skoog growth medium for plants. In this lab session you will learn the basics of plant tissue culture growth medium, its components, methodology of preparation and points of attention while preparing the medium. Introduction. Can you imagine in vitro culturing without using media? No way. Right? But, why? The answer to the question is simple, because plants need nutrients for their growth and survival, like all other organisms. In a natural environment, they fulfill their needs by getting it through the atmosphere, soil, and by associating with other organisms. However, when plants are cultured inside the lab, the support and nutrients for their growth are provided to the plants through media. In the theory section, you have already learned about the various types of plant tissue culture growth medium which is used to culture in vitro plants. You have also studied about the purposes served by different culture mediums, and how to select the right media for your plant in just 5 steps. You must know, of all the media, the Murashige and Skoog (MS) medium is one of the most extensively used in plant tissue culture labs worldwide. MS medium was invented by plant scientists Toshio Murashige and Folke K. Skoog in 1962 during Murashige's search for a new plant growth regulator. Murashige and Skoog medium, MS, was originally formulated by Murashige and Skoog in 1962 to optimize tobacco callus bioassay system for facilitating the study of cytokinins. Since then, it is widely used for micropropagation, organ culture, callus culture, and suspension culture. The formulation is a nutrient blend of inorganic salts, vitamins, and amino acid. So, what is its recipe? What concentration of nutrients is required for the media? In this virtual lab session, you will answer all of the above questions, with a short description of components of the media. You will also find a handy chart that you can keep with you while preparing the media in your lab. Principle. The success of getting callus and morphogenesis of plant tissues and applications of in vitro methods is largely governed by the better understanding of nutritional requirements of the cultured cells and tissue composition of the cultured media. Although the basic requirements of the cultured plants are similar to that of whole plants, the nutritional requirement consists of inorganic salts, carbon, and energy source, vitamins, and phytohormones, other components including organic nitrogen compound, organic acids, and complex substances. In practice, nutritional components promoting optimal growth of tissue under laboratory conditions may vary with respect to the particular species. Media composition was therefore formulated considering specific requirements of particular system. Media components. One of the most important factors governing the growth and morphogenesis of plant tissues in culture is the composition of the culture medium. The basic nutrient requirements of cultured plant cells are very similar to those of whole plant. Plant tissue and cell culture media are generally made up of some or all of the following components. First, macronutrients. Second, micronutrients. Third, vitamins. Fourth, amino acids or other nitrogen supplements. Fifth, sugars, and/or other undefined organic supplements. Sixth, solidifying agents or support systems. And seventh, plant growth regulators. Several media formulations are commonly used for the majority of all cell and tissue culture work. Macronutrients. The macronutrients provide the 6 major elements: nitrogen, N, phosphorus, P, potassium, K, calcium, Ca, magnesium, Mg, and sulfur, S, required for plant cell or tissue growth. These elements are generally added in the medium in its inorganic salt form. The optimum concentration of each nutrient for achieving maximum growth rates varies considerably among species. Culture media should contain at least 25 to 60 millimolar of inorganic nitrogen for adequate plant cell growth. Plant cells may grow on nitrates alone, but considerably better results are obtained when the medium contains both a nitrate and ammonium nitrogen source. Certain species require ammonium or another source of reduced nitrogen for cell growth to occur. Nitrates are usually supplied in the range of 20 to 25 millimolar, typical ammonium concentration range between 2 to 20 millimolar. However, ammonium concentration in excess of 8 millimolar may be deleterious to cell growth of certain species. Potassium is required for cell growth of most plant species. Most media contain potassium, in the nitrate or chloride form, at concentration of 20 to 30 millimolar. The optimum concentration of phosphorus, magnesium, sulfur, and calcium range from 1 to 3 millimolar when all other requirements for cell growth are satisfied. Higher concentration of these nutrients may be required if deficiencies in other nutrients exist. Micronutrients. The essential micronutrients for plant cell and tissue growth include iron, Fe, manganese, Mn, zinc, Zn, boron, B, copper, Cu, and molybdenum, Mo. Chelated forms of iron and zinc are commonly used in preparing culture media. Iron may be the most critical of all the micronutrients. Iron citrate and tartrate may be used in culture media, but these compounds are difficult to dissolve and frequently precipitate after media is prepared. Murashige and Skoog used an ethylene diamine tetraacetic acid, EDTA, iron chelate to bypass this problem. Cobalt and iodine may also be added to certain media, but strict cell growth requirements for these elements have not been established. Sodium, Na, and chlorine, Cl, are also used in some media but are not essential for cell growth. Copper and cobalt are normally added to culture media at concentration of 0.1 micromolar, iron and molybdenum at 1 micromolar, iodine at 5 micromolar, zinc at 5 to 30 micromolar, manganese at 20 to 90 micromolar, and boron at 25 to 100 micromolar. Sugars, amino acids and other nitrogen supplements. Carbon and energy source. The preferred carbohydrate in plant cell culture media is sucrose. Sucrose concentration of culture media normally ranges between 2 to 3%. Vitamins. When plant cells and tissues are grown in vitro, some vitamins may become limiting factors for cell growth. The vitamins most frequently used in cell and tissue culture media include thiamine, B1, nicotinic acid, pyridoxine, B6, and myo-inositol. Thiamine is normally used at concentration ranging from 0.1 to 10.0 mg per liter. Nicotinic acid is normally used at concentration of 0.1 to 5.0 mg per liter. Pyridoxine is used at 0.1 to 10.0 mg per liter. Myo-inositol is commonly included in many vitamin stock solutions. Although it is a carbohydrate, not a vitamin, it has been shown to stimulate growth in certain cell cultures. Myo-inositol stimulates cell growth in most species. Myo-inositol is generally used in plant cell and tissue culture media at concentration of 50 to 5,000 mg per liter. Amino acids or other nitrogen supplements. Although cultured cells are normally capable of synthesizing all of the required amino acids, the addition of certain amino acids or amino acid mixtures may be used to further stimulate cell growth. Amino acids provide plant cells with an immediately available source of nitrogen, which generally can be taken up by the cells more rapidly than inorganic nitrogen. Examples of amino acids included in culture media to enhance cell growth are glycine at 2 mg per liter, glutamine up to 8 mg per liter, asparagine at 100 mg per liter, L-arginine and cysteine at 10 mg per liter, and L-tyrosine at 100 mg per liter. Tyrosine has been used to stimulate morphogenesis in cell cultures but should only be used in an agar medium. Undefined organic supplements. Addition of a wide variety of organic extracts to culture media often results in favorable tissue response. Supplements that have been tested include protein hydrolysates, coconut milk, yeast extracts, malt extracts, ground banana, orange juice, and tomato juice. The addition of activated charcoal to culture media may have a beneficial effect. The stimulation of cell growth by activated charcoal is generally attributed to its ability to bind to toxic phenolic compounds produced during culture. Solidifying agents and plant growth regulators. Agar is the most commonly used gelling agent for preparing semi-solid and solid plant tissue culture media. Agar has several advantages over other gelling agents. First, when agar is mixed with water, it forms a gel that melts at approximately 60 degrees to 100 degrees Celsius and solidifies at approximately 45 degrees Celsius. Thus, agar gels are stable at all feasible incubation temperatures. Additionally, agar gels do not react with media constituents and are not digested by plant enzymes. The agar concentration commonly used in plant cell culture media ranges between 0.5 to 1.0%. Plant growth regulators. Four broad classes of growth regulators are important in plant tissue culture: the auxins, cytokinin, gibberellins, and abscisic acid. The auxins commonly used in plant tissue culture media are indole 3-acetic acid, IAA, indole 3-butyric acid, IBA, 2,4-dichlorophenolic, acetic acid, 2,4-D, and 1-naphthalneacetic acid, NAA. Auxins generally stimulate cell division, cell elongation, callus induction, induction of somatic embryogenesis and in vitro root induction. BAP and chitine are two important cytokinin used in plant tissue culture medium. Cytokinin stimulate cell division, morphogenesis and de novo bud proliferation. 2 mg per liter concentration of cytokinin is generally recommended. The concentration of auxins and cytokinins are equally as important as their ratio. Gibberellins, GA3, and abscisic acid, ABA, are two other growth regulators occasionally used in culture media. Glassware requirements. Following glassware are required for MS medium preparation: conical flask, test tubes, measuring cylinder, round bottom flask, and culture bottles. Plasticware requirements. Following plasticware are required: micropipettes, microcentrifuge tube, and polypropylene beaker. Chemical requirements. Following chemicals are required for preparation of MS medium: MS stock solution A, of macronutrients; MS stock solution B, of micronutrients; MS stock solution C, of iron and chelating agents; and MS stock solution D, of vitamins and organic supplements. Beside this, agar powder, sucrose and ethyl alcohol is also required. Other chemical required are: auxin stock solution, cytokinin stock solution, pH buffer solution, chemical sterilizers, like mercuric chloride and sodium hypochlorite. Equipment requirement. Following equipments are required: autoclave, pH meter, weighing balance, and microwave oven. Reagent preparation. Preparation of MS stock solution is the most basic and important step in the plant tissue culture medium preparation. To prepare any basal medium, including MS medium, the ingredients like macronutrients, micronutrients, iron EDTA, and vitamins are common. So, in order to prepare any basal medium, stock preparation is crucial step. To avoid any kind of error in weighing and to save the time, it is better to prepare stock solutions of standard concentration and use whenever it is needed to prepare the medium. The components of different stock is mentioned in this table. It's completely on your choice that how much volume of stock, and how many different stocks of inorganic and organic supplements you prepare. In this table, the main constituents of macronutrients, stock A, and micronutrients, stock B, their stock concentration, their final required concentration in 1 liter medium, and the volume to be taken to prepare 1 liter of medium, is given. You can pause the video and note it down for your future reference. Similarly, the constituents of stock C, iron EDTA, and stock D, vitamins and organic supplements, their stock concentration, their final required concentration in 1 liter medium, and the volume to be taken to prepare 1 liter of medium, is given. You can pause the video and note it down for your future reference. MS media preparation. Culture media are largely responsible for the in vitro growth and morphogenesis of plant tissue. Murashige and Skoog medium is the most suitable and commonly used of basic tissue culture media. To prepare 1 liter of MS media, first take the double distilled water in a container such that it is 40 to 50% of the final volume of media to be prepared. To make this process easy and fast, we prepare stock solutions. These are the 6 stock solutions used in the MS media preparation. Add the required quantity of stock solution to obtain the specific concentration of each nutrients. Macronutrients stock solution, which contains major element for growth and development of in vitro plant. To make 1 liter of MS media, we will be taking 50 ml of the 10x concentration of macronutrients stock. We prepared 100x concentration of minor nutrients. From this we had to take 2.5 ml of stock solution. Add 100x concentration of 1 ml of micronutrient stock. Prepared 50x concentration of iron stock. From this add to 2.5 ml of stock solution. We will be taking 2.5 ml of 100x concentration of potassium iodide stock. Add 1 ml of the 100x concentration of vitamins and amino acid stock solution. Media should contain 3% of sucrose. To prepare 1 liter of MS media, add 30 grams of sucrose. Dissolve it properly. Adjust the pH of the medium from 5.6 to 5.8 by using a 1 normal NaOH or HCl. Make up the volume to 1000 ml by using distilled water. And 8% of agar. For 1 liter of media preparation, add 8 grams of agar. Melt the agar in microwave oven. If we can add liquid hormones into media that is essential for plant growth. These are the commonly used hormones in tissue culture media. To make 1 ml of stock solution, dissolve 0.5 milligrams of required hormone in absolute ethanol or NaO. Distribute approximate 20 ml of media into each culture bottles. Close the lid of the bottle. Autoclave the culture media for 20 to 30 minutes at 121 degrees Celsius. The following precautions needs to be taken while preparing the medium. All stock solution should be transparent, and without any kind of precipitation. Stock solution C should be stored in amber color bottles. Stock solutions should be kept at 4 degrees Celsius. These solutions should be stored for a limited period of times. Stock solutions should be taken out of refrigerator well in advance. It should be ensured that sufficient glassware and their stopper are available. Do calculations of stock requirement carefully well in advance. pH adjustment is essentially required. Generally, acidic medium does not get solidify. Agar should be dissolved in lukewarm water, otherwise it will form aggregates. Proper autoclaving is essential to avoid any kind of contamination. Answer the following questions to check your understanding of this virtual lab session. Question 1. Which of the following is used as macronutrient in MS medium? A. Nitrogen. B. Molybdenum. C. Copper. D. Both A and B. Question 2. Which of the following is not the component of MS medium? A. Agar. B. Nitrogen. C. Silicon. D. Aminositol. Question 3. What should be the pH of MS medium? A. 5.8. B. 6.8. C. 7.0. D. 7.4. Question 4. What is the advantages of preparing stock solutions in MS medium? Question 5. Compare and contrast the MS medium for plant tissue culture and nutrient agar for bacterial growth. Question 6. Find out the stock concentration of following chemicals of MS medium. This is a list of some books and literature for further deep study. I remain most grateful for the rapt attention with which you sat through this virtual lab. I'm confident that you've learned something from it. You can contact me for further queries on below email address. Thank you very much.